Hello, everyone. So we are here for the Deep Sea Biology seminar series, and the second one of this year. And today we are hosting two speakers that going to talk about the use of a very interesting method, 3D photogrammetry, on different deep sea ecosystems. So we will first have a presentation by Loic Van Audenhaege on the use of 3D photogrammetry in hydrothermal vent, and then a presentation by Ana-Belen Yanes Suarez on the use of this method in deep sea coral reef. So Loic, if you want to start, the room is yours.
Thank you very much, Pierre, and thank you everyone for joining today. So my presentation will be split with a small presentation first, a small introduction on the methodology, how do we produce 3D models. So I will share my screen. This is just a presentation I'm reusing. Can you all see my screen? I guess so, because we made the test. So the goal of photogrammetry is to create three-dimensional coordinate measuring technique that uses photograph as the fundamental medium for measurement. So very briefly, what you do, you take picture from different angle of view, picture that are overlapping, right? And you put them in the magic software that will basically detect common feature among your images. So for example, those points, color points on the square, on this cube, sorry. The software will recompute the original position of the camera, and then it will be able to triangulate the position of the original point. So recreate basically a point cloud with a virtual environment on your computer. Then densify the points, and then you create a mesh. And this is what we both use, myself and Anna. Right, that's it for the principle. If you have any question, of course, we can talk about that later. And let's move on the ecology side. Monitoring ecological dynamics on complex hydrothermal structures. So this work is my second chapter of my PhD that I was doing at Ephemer. Now I'm working at NOC on a flatter abyssal plane, but I still like to use photogrammetry. So temporary dynamics in ecology, such as community maintenance, natural recovery, is important to develop fundamental knowledge in ecology and to develop conservation baseline. Especially, for example, for deep sea hydrothermal vents that are now the potential future targets of deep sea mining. Deep sea hydrothermal vents are dominated by large and abundant specialists fueled by chemosynthetic compounds. And they exhibit patchy aggregation or assemblages that segregate along the vent fluid dilution gradient according to their tolerance to toxicity, trophic needs, and so on. So that makes these communities, because they're quite patchy and we can see some large megafauna, as you can see here on the screen, it makes them ideal, imagery makes them ideal for the monitoring, basically. So a bit of state of the art in terms of monitoring at hydrothermal vents. There have been a few studies on community dynamics at active edifices, which are those vertical structures that concentrate vent outflows, such as, for example, different studies such as post-erosion studies in the Pacific, which is characterized by intermediate to fast spreading ridges where eruptions occurs every 10 to 20 years. And we can observe a reset and a rapid recovery of the biology there. For the past decades, there have been more studies in the mid-Atlantic ridge, which is a slow spreading ridge. And therefore, eruptions are very, very rare. We talk about thousands of years in terms of frequency. So we've observed stability. However, the mechanisms that maintain the stability have been poorly resolved today because of the use of 2D images and the use of opportunistic image sets. So the goal of this research is to describe polyannual dynamics of an edifice assemblage from large to fine scale under a square meter by monitoring their succession and scale of maintenance using centimeter resolution methods, for which photogrammetry is a suitable approach. So to do so, we will focus on the Eiffel Tower edifice at the Lucky Strike Van Field here, which is the site for the observatory, the Emzo Azores Observatory. So scientists from IFMR are coming back every year on the sites to collect sample, and we take advantage of those cruises to acquire images. So Lucky Strike is located on the mid-Atlantic ridge close to the Azores Islands, as you can see here, sorry. Right, so now photogrammetry. So we use the ROV Victor 6000 to do vertical transects all around the edifice of Eiffel Tower. So we acquired a 360 photogrammetry model with an area of 450 square meters. So we did that in 2015, 2018, and 2020. We try to georeference those models, and as you can see here, we can really zoom in the details of the model to actually distinct the different substrata and assemblages. So this is on the 3D model, and because we managed to georeference them with an accuracy of more or less 1 to 5 centimeters, we were able to create a very high precision time series. As you can see here, those are the same area. Okay, so the assemblages and substrata we find at Eiffel Tower are basically large mussels, Bathymodelus azoricus, medium, smaller mussel patches, anemones, empty shell patches, black and white substrata, which describe active venting, and microbiomats that cover the assemblages. We also find vent fluid exits, of course, and topographic changes. Sorry, sorry, I misled myself. So basically, we annotated those substrata and vent fluid exits and topographic changes over the 3 edifices. So we annotated 4,300 polygons, and because we had good enough registration, let's say, we were able to actually acquire a time series for each polygon. That's quite important for the next slide. So I will jump straight into the results. I won't spend too much time on methodology, but I try to explain all the figures as I go along the results. So first, this is a successional scheme. So basically what it shows you is each box represents an assemblage. Don't look at the size of the box. It's not to scale with the area covered by the assemblage. Those boxes are organized according to the known distribution of substrata and assemblages. So for example, black and white material closer to venting activity, large, smaller mussels, and then in periphery, empty shells, zoanthid, and so on. You can see the microbiomats occupy a wide array of condition and niche, temperature niche. In these boxes, the left number shows you the total cover that was counted in 2015, and in the right number is the total cover in 2020. So in the periphery, we can see that assemblages were quite stable. We observed slight changes for the mussel Bathymodelus azoricus of around 1 to 2 square meters per year, so from 2015 to 2020. Can you see my mouse, Pierre, by the way? Yes, okay, it's good. Wasn't sure. And but compared to this is not really significant compared to the high decline we observed for microbiomats. Nearly 50% of the cover disappeared in 2020. So something that is quite interesting as well in this graph is these arrows for which their width shows you which area converts from one assemblage or substratum to another, for example. And you can see that, of course, they are still transferred between one assemblage and the other, but that basically explains you that this stability is probably kind of a climax that we observed also in the past, that is maintained because of dynamic equilibrium between assemblages and substrata. Something that is interesting as well with these arrows, if we summarize them even more, is that they take kind of a linear and unidirectional scheme, let's say. So you have, for example, A will never transit to D directly, which is something we have, however, observed in Juan de Fuca on the Pacific ridge. This kind of scheme is more, it has been reported before in ecology in general, and is more relevant for communities that undergo chronic disturbance. So here, for Eiffel Tower, the high stability may be explained actually because we have very local disturbance, most likely, to which communities recover very quickly or can cope with, actually. But we try to go explain that with other results. So habitat change now. So you can see here a map of Eiffel Tower looking from above. And all those symbols represent different fluid outflows that were observed in the 3D models and the videos. First of all, you can see that they concentrate well within distinct areas that I delineated with dashed lines. And second, you can see that those areas are very stable through time. So from 2015, 2018 to 2020. The only thing that you can notice is probably that those vent outflows seems to rearrange, but at very small scales, which is known. So outflow will open and close very dynamically, but at less than a meter scale, let's say, probably at a decimeter scale. In terms of topographic changes, we observe mostly the effect of, we observe mostly in venting areas, accretion of materials. So for example, this flange that seems to be growing frontward. And you can see as well that mussels here seems to colonize following this gradual advancement of the flange. So it's maybe due to mobility, as we've observed that with the deep sea observatory mussels in aggregation are capable of moving of around 10 centimeters per year. All right, so here what I want to show you is that the habitat is relatively stable in general, except within those venting areas. And that might explain as well why we observe such a high stability in the previous results. Okay, let's go for a more difficult result. So now what we want to do is try to map how these assemblages change over time, but to map those changes as well on Eiffel Tower. The problem is that we annotated polygons which have a variable surface and want to plot them in a standardized way over the map of Eiffel Tower. The problem, so what we have to do is to aggregate them in tiles of around 1.7 square meters based on their proximity, as you can see here. Now with this, it's much more easy to create a symbol that will represent, for example, how much cover a single assemblage has changed in one single tile, and to plot that onto the map. So this is what we try to represent with these circles. You can see here, for example, in white, the cover that's cover that undergo no change, and in green, the cover that undergo a change. And the radius of the circle is proportional to the cover, the actual cover it represents, right? And we can make it even more, a little bit more complicated and informative, meaning that the change can be disentangled in different parameters. either if we have a net gain or a net loss. And the remaining cover changing is basically polygons that compensate each other. So for example, in 2015, the loss will be compensated by the gain in 2020, if that makes sense. And again, the radius will explain the cover represented by each of these parameters. We can then plot that on the map and assess changes over space. Right, this is what I've tried to do for large muscles on the left, medium muscles, small muscles, and microbial mats. So first of all, you can see that large muscles, the distribution along the vent fluid dilution gradient is quite respected. So large muscles seems to cluster mostly in the venting area. Medium muscles are present in the venting area, but mostly outside of the venting area. And small muscles seem to position on top of the summit of Eiffel Tower, so they are much further away from venting exposure. And microbial mats seem to cluster in different areas, as you can see by the presence of circle in venting area and outside. So now about the change, we try to analyze the colors of the circle. Except for this area where we see an increase of large muscle, the vent muscle seems to be either unchanged or with the white color or with the green color. It seems like there is a null change, meaning that they may actually either grow very quickly or displace to maintain their position in adequate exposure to the vent fluid, right? For medium muscles, except in venting areas, we observe an overall stability on the edifice, as shown by this white circle, except in this very specific patch, which transition from medium to small muscles for a unknown reason. And for microbial mats, except in venting areas, it seems that the decline occurred mostly on the edifice. Right. So summary of results, we observe contrasting dynamics between microbial mats and vent muscles. So we observe decline of microbial mats, and that may be due to a plumbing event that happened in the depth of the hydrothermal plumbing system, let's say, and that was detected in 2015. So that could have changed venting conditions to which microbial mats are sensitive, but only with imagery we were not able to resolve that, let's say, because the visual distribution of vent outflow doesn't seem to change, except probably the composition. We observed the high stability of vent muscles with extent or knowledge of 5 years to 25 years based on previous data that Cuvillier et al. Used with 2D images. So since 1994, it's quite a long time, let's say, for a very stable population. And this is explained by the low regime of disturbance. So those muscles are able for 25 years probably to maintain themselves in a climax state. Most of the disturbance occurs in the venting area because of fine changes of growth of let's say, material or repositioning of vent outflow. And we suggest the role of mobility for muscles that may displace to cope with displacement actually of the vent outflows. Right, and I will try to integrate those results. So in terms of ecology, compared to the first and pioneering Pacific Ocean studies, the vent ecosystem is not necessarily described by rapid and intense changes, as it was observed on the Rouen-Desfoucas Ridge or in the East Pacific Rise. And even here, we observe that it can reach a climate stage in the Mid-Atlantic Ridge. So other questions can arise from that. How much time, what time scale do we need to reach such a climate stage? Is it like centuries? Is it thousands of years? Considering that an eruption is likely to occur after a few thousand years, this question is unanswered because we never observed such events to date in slow spreading ridges. are these communities important for population maintenance? Because this climate has subsisted over a long time. Eiffel Tower harbors quite an extensive community. Could this population of mussel, because of this stability, be important to actually supply larvae at a regional scale? In terms of this mining, so we demonstrated that photogrammetry is a reliable method to quantify precise indicators of change and to establish ecological baseline that might be important for before and after a collection test occurs. So we could be able to detect even change that occur at, let's say, a meter scale, not even the edifice scale, which is easier, let's say, to observe with 2D images, but we can go deeper with 3D photogrammetry. And the last question that subsists is, because we observe such high stability, are we not sure that vent organisms are actually adapted to naturally recover after an intense disturbance? Indeed, they may not have evolved and developed the traits to cope with intense disturbance, such as it has been hypothesized in the Pacific Ocean because of the high frequency of eruptions. So thank you for listening to this presentation. If you want to go further, this work has been published in limnology and oceanography, following this QR code. And if you want to enjoy a nice 360 dive on your phone on YouTube, you can follow this QR code as well. And feel free to ask any question you want. Thank you very much.
Thanks a lot, Loic. It was a very nice talk. We have time for a few questions before Anna's presentation. So if anyone have something to ask to Loic. If not yet, I have a few questions, so people can think about.
Or even later after Anna's presentation.
Yeah, of course. I was wondering, so you were with your 3D model, you were comparing 3 different years, so 2015, 2017, and 2020. Where the model and the imagery were they taken at the same period of the year? I guess it's cruise dependent, but like were they the same month or were they at different time?
As far as I remember, it's ranging between April and July. So the cruise called Montmartre, it's usually happening before summer or in summer. But unfortunately, yes, it is cruise dependent, and you could have a seasonal effect probably, but although I have no hypothesis how seasonal effect could, maybe it could have an effect on smaller mussels, because indeed we know that they actually feed also on particles that come from that just sediments
sinking,
from
yeah. I think for the mussel from Lucky Strike, they shown for Lucky Strike, but also on the Gulf of Mexico, that the reproductive cycle were influenced by the seasonality, and the input was probably like the variation of the photosynthetic primary production sinking down from the surface. So I was wondering like if it could have an effect on the assemblage, and what about your the period of the year where your model were created. I was also, and there's something I wasn't sure I understand properly. So you said that you had a decline for the microbial mat overall. Was it because you compared 3 years, so was it variable between like you had a decline and then an increase, or was it like a kind of like a constant decline between those 5 years?
So it depends on the scale you look. If you look at the edifice in general, the decline was gradual, so it was a trend, let's say. We've seen with Tempo, so the time-lapse camera that was deployed right here. I'm just going back right here, which was my first PhD chapter. We have observed that if you look at small scale and at monthly period, microbiomass seems to actually reposition quite dynamically of a space. So it might not be such a relevant scale to look for microbiomass, but yeah, in overall over the edifice, it's more like a trend, let's say, a climbing trend.
I think Verena Tunnicliffe have a question for Loic.
Yes, Loic, that was a lovely presentation. Thank you very much.
Thank you.
And I really strongly agree with your hypothesis that that not all vent organisms or areas are adaptable to change. I mean, even on Juan de Fuca Ridge, there are different vent sites that have different eruptive frequencies. So for example, on axial seamount, we have a reduced diversity, and there are definitely species that are really highly adapted to dealing with change. And so recovery can happen quickly with some species, and that's the same on land. After a fire, you will find that there are some species that get in very quickly, and other species that some of them just never get back. So that concept that we need to look perhaps even beyond just a few dominant species, and maybe just thinking, I'm just wondering if anybody is mapping some of the other species that the kinds of species people rarely study at hydrothermal vents because they haven't got a broad distribution or they're not dominating.
One of the issue if you map all the species, so for example, let's say crabs, is that they're highly mobile, and they hide in muscle assemblage. So it's very difficult to extract a pattern from that, let's say. And otherwise, the species you can see, so for example, at Eiffel Tower, it's known at other vent edifice, but there is a small patch of vent gastropods, but it's really like one patch of less than 50 square centimeters. So it's not really robust in terms of science, let's say, it's in terms of statistic, let's say. However, I agree with you, because here we only consider a small set of the community, and I mean, much less is known, let's say, on the recovery of those communities. There is this paper from Julien Marticorena here. So they experience a clearance on muscle assemblage, and it seems to be, there is kind of a successional recovery, a successional pattern, sorry, that happens when the assemblage is recovering, although muscles don't seem to recover after 5 years, which is quite contrasting compared to Pacific studies, let's say. Otherwise, I think what could give a better picture is definitely macrofauna. So for example, the work of Moulineau, for example, which is very interesting. And the fact that, for example, even if some papers mention that recovery and was reached after a few years, in 2020, she published a paper after a 20-year time series, I think, with the macrofauna, stating that recovery might not be reached after 20 years, for example. So, but that's because if you do an experiment for macrofauna, you can collect your sample back. Here we can't really be, let's say, invasive, which is the beauty of imagery, but we only see what we see, let's say.
No, that's great. Thanks a lot.
Thank you.
Also able to ask more questions after the second talk.
Yeah,
So
there's my
think,
email as well. So
yeah.
if I will stop sharing my screen.
Thanks, Loic. So
Thank you.
Anna, if you want to start.
Sure, I will share my screen. Thank you so much for the lovely presentation, Loic. Let's start. All right, here we go. Sure. I will have to do it again. We're gonna share the whole desktop. Okay, share. All right, can you see my screen? Okay, just let me do something here. Just give me a second because I cannot see. Zoom just closed. Oh. Is acting very weird. Okay. I'm gonna have to stop sharing because this is just not working. So now share. Oh. All right, there we go. You're sharing a screen. Yes, I'm sharing a screen, but I cannot see you. Oh my God. So. Maybe, wait. Please just stay like that.
You were sharing the PowerPoint, so for me.
Yeah, this is what happening if I share, for example, this. You can see now.
Now we are seeing a table from a paper.
Yeah, this is not really working very well. But maybe if I do this.
I think the previous one was okay, and you can just start slideshow now.
Yeah, the problem is I cannot see the screen with you. So I will have to reconnect and connect again.
Okay, well, just do it quickly.
Okay, we have to leave the.
And yeah, no worries. Loic, if anyone else have a question for Loic while we were waiting for Anna, technical issue.
Yeah, I'm still here.
Yeah, about the idea of looking at other animal with photogrammetry, like you say the crab, but I was also thinking of the shrimp, because I think there is a shrimp assemblage also in the Lucky Strike mound.
That is a good point. If you can rebuild it, there's Capellinius. For example, if you want to rebuild something like with the Remicaries large densities, is very hard to rebuild because they move all the time.
Yeah, yeah, too mobile. We can see we can see your presentation, Anna. Get the presentation in full screen, but we are just not hearing you. I don't know if you're muted. I think you're muted.
Right, I'm so sorry for all this.
It's okay.
But now it's working, and so let's
Yeah,
start with
we
it.
can hear you. We can see the
Great.
screen. It's perfect.
So thank you so much for your time and for your patience. And I appreciate very much that you're here. So today I'm going to talk about one of my chapters, habitat selection and niche differentiation of cold water corals and sponges and deep sea vertical walls. So first, I want to share with you this video of a vertical wall. Vertical walls are characterized by hard substrate and low sedimentation rates. This can provide an excellent habitat for corals and sponges to occupy, and these species that are vulnerable marine ecosystem indicators create extra complexity and habitat for other organisms such as crabs, elasmobranchs, fish, and more. So the biodiversity is increased. Now, here you can see a vertical wall. Vertical walls are protected from some human impacts such as bottom trawling fisheries. They are not target of that, so the species of corals and sponges that live in these vertical walls can be protected from these impacts and serve as source of larvae to recolonize the flat terrain. And so now that you know how important these vertical walls can be, I have to mention that not all of them are the same. So although there are some vertical walls that have high diversity and dense communities, there are others that are just bare rock. However, the habitat characteristics that favor the presence of cold water corals and sponges within these vertical walls remain poorly understood. So here I want to show you this image of a vertical wall and this tree, this pine tree. What does it have to do with each other? They are similar because they have heterogeneity. What I mean by that is that they have different areas, different microhabitats that the species use and can use in different ways in order to coexist. This is through a very important ecological principle that's called niche differentiation. So as the species of webblers here use this tree differently, the species of corals and sponges in vertical walls might also do the same with heterogeneous vertical walls. So highly heterogeneous structure provide more microhabitats to be used by diverse species. In complex vertical walls, species might inhabit different sites according to their preferences. The objective of this study is to identify habitat selection and niche differentiation between species of corals and sponges in vertical walls. The area of study is the Charlie Gibbs fracture zone. This is an important geological and topographic feature that divides the Mid-Atlantic Ridge horizontally. It's one of the first marine protected areas in international waters. Currently, the southern portion, here in green, is fully protected, but the northern portion, now in red, only the water column is considered a marine protected area, not the benthic communities. Here is where our study took place. And here you can see three of the transects that I used to get all the data that I needed for this study. The data was collected during the tectonic ocean spreading at the Charlie Gibbs fracture zone expedition in 2018. So my questions are, do corals and sponges show habitat selection for specific conditions? Which variables are relevant for corals and sponges habitat selection? And is there niche differentiation between species in vertical walls at fine scale? So as you hear me, I mention scale. It's because in my methods, scale is going to be very important. I'm going to study these questions through terrain variables. So originally and traditionally in the deep sea, because how hard it is collecting fine scale terrain variables, we have used large and mesoscale that can be used to develop coarse predictive habitat models, identify potential areas where species might be present. So they are important. However, in order to understand these important ecological questions such as niche differentiation and habitat selection, we need to go a bit closer. So I'm going to use fine scale terrain variables under 1 meter to identify the different important biologic and abiotic processes at a scale experienced by these benthic organisms. How can we measure this? So here we can measure this by using structure from motion photogrammetry techniques that allow me to create these 3D models based on real image captured by ROVs. These 3D reconstructions allow us to explore the deep sea as if we will be diving in there, and create maps where you can identify and geotag each organism where they are. So that's what I did, and I create 3 vertical walls for a total of 569 square meters. And here I want to share one of them. So please pay attention to the level of definition of the terrain that we can achieve. So you can see all the crevices and the rock structure, and also the different orientation that this wall has. It doesn't have only one orientation if you pay attention to that. Also, I decided to annotate the species here in the 3-dimensional space, so we know where they are located exactly. Now what you are looking at are the terrain variables derived from clouds, from these point clouds created from these images and coordinates. So I'm going to talk more about which variables I use here. So first of all, I use 6 scales, 2, 5, 10, 20, 30, and 50 centimeters. And the variables I look at were slope, rawness, mean curvature, verticality, and orientation, the terrain variables I look at. Why? All these terrain variables seem to influence species presence, but it is because they are proxies of community structuring factors that are harder to measure in the deep sea, such as sedimentation rates, local currents, and nutrient availability. I also use depth because we know this is an important variable. In the morph species I studied were Solenosmilia, some species of the family Paragorgidae, Herwigia, Geodia, and globular sponge. I use multivariate statistics, in this case, ecological niche factor analysis, that help me to understand where the organisms are established, so the niche, what they are searching for, so the habitat selection, and identify which variables are the most important for each organism. Then I use outline mean index to understand if there is niche differentiation between and among morph species. Morph species, and also the tolerance of the species to environmental change. To see if what I found was significant, I used permutation tests, in this case, Monte Carlo with 999 permutations. Now we're gonna dive into the results. So to remind you my questions, do corals and sponge show habitat selection for specific conditions? Which variables are relevant? And is there niche differentiation? For all of them, we find an answer. So each morph species shows significant habitat selection for specific conditions, different from the average habitat available to them. They all had a narrow niche breadth, which indicate that they have low tolerance to environmental variability. The most relevant variables were verticality, mean curvature, orientation, and the scale that was the most important was 0.5 meters. There were different levels of niche differentiation between corals and sponges. And let's look at that a bit deeper. So here you can see a representation of the average habitat conditions and the niche of one species. So if you pay attention to the larger polygon that looks almost like a circle, this is the average habitat available to the species. And then the darker polygon, dark gray, this is actually the niche of the species. So in the x-axis, here, you can see how the niche goes very far away from the mean habitat conditions that are available to it, meaning that this species has a highly selective behavior for specific variables. And in this case, the most important was isness for Solenosmilia, which makes sense because isness is orientation, and orientation is related with currents. So currents might be something very important for this species. Then we look at Herwigia. Currents, a very fine scale I'm talking about. So Herwigia, they show the most strong habitats. The variable that drives the marginality of this species, as you can see here, is mean curvature. And mean curvature was related with overhangs across the vertical wall. So living in these overhangs might give this species some advantage. And to look deeper into this species, we also should look at the at the vertical axis, where we can see that both of them are narrow, indicating lower tolerance to environmental variability. Also, it's interesting to see that the niche of Solenosmilia is a lot smaller than the one of Herwichia, but that we can see better here. With the Olaye index analysis, I was able to find that Solenosmilia has this smaller niche, and although there is some overlap with the niche of Herwichia, most of the niche is separated. But what are these variables that are driving that? Again, is the orientation, in this case, isness for Solenosmilia, and for Herwichia, it is preference for overhangs in the vertical wall. Now, in the outer wall, I had the presence of Solenosmilia, Geodia, and globular sponge. And what happened is that Solenosmilia, as you can see, this circle here is completely separate from the circles that represent the niche of the sponges, and indicating that there is not a niche overlapping windows. And what was driving that was, again, the orientation and also rawness. And for the case of the other two species, there was a lot of overlap between these two species, which makes sense because we couldn't go to a very high taxonomic level with this globular sponge, as you might assume. That's why I give it this name. And these species are very similarly morphologically. Jod is also round, as you can see. So in the case of the last wall, we found Paragorgia in this Geodia and globular sponge. And again, Paragorgia, the coral, the soft coral in this case, show a higher niche differentiation, niche separation from the niches of the sponges studied. And interestingly here, it was, well, C, that means depth here, was what drive this separation. But yeah, you might think, yes, we know depth is important for deep sea species because it is collinear with other parameters such as temperature and oxygen in some cases. But in this particular case, we're talking about very fine scales. And one thing that we could notice is that the shallower portions of this reconstructed vertical wall were less exposed to the bottom. So there was less sediment accumulation. And see, you can observe here a yellow coral here living in this area, while in the deeper areas that were closer to the bottom, there was more sediment accumulation. And so although sponges are also sensitive to sediment accumulation, they have more adaptations that can help them to survive. And so that might be the explanation to this observation. So the take-home message is that the habitat selected by the morphic species was significantly different to the average habitat available. So they do have a habitat selection. And there was some level of niche differentiation, more between sponges and corals than among the same between the same sponges, similar sponges. And that is interesting because that shows that this niche partitioning is happening at a certain level in vertical walls. And even only using terrain variables, we were able to detect that. So this niche partitioning indicates that these competitive sessile filter feeders might be able to cohabitate in these complex vertical walls by selecting slightly different habitats. So you might remember this image with a wall full of organisms and another bedrock. So the morphic species selection of different fine scale terrain variables can help to explain how vertical walls can be more or less suitable for corals and sponges. If we think about this pine tree, we see again that vertical walls that might have more heterogeneity in terms of roughness, but also different orientations might provide more areas for different organisms to partition their habitat through niche differentiation and be more biodiverse and full of life. So I know the ecological findings are very relevant for me, but people always want to hear a bit more about the potential applicability of all this. So it is important to mention that in recent years, there is a bigger trust in restoration of these sea corals because now we have the technology. And however, the effectivity of this restoration project will vary depending on the species, but also the sites that are chosen for restoration. If we use fine scale, if we can access the fine scale habitat selection of these species, we might be able to improve our ability to have a project of restoration that works better. So thank you so much for your attention. And if you want to hear more about my research, please scan this QR code and follow me. And also check the website of our lab for the oceans at the Marine Institute at Memorial University. And I'm very happy to hear your questions.
Thanks a lot, Anna. That was a really great talk. I have a
Thank
lot
you
of
so much.
questions, but maybe other people in the room have also some questions.
Thank you. Thank you for the patience with the presentation. It's always
No
hard.
worries. Like, and for the room, for the people that arrive later, we made a test before, so even with that,
Yes.
we like, as everyone
Problems
knows,
never
there are
happen
some
when they have to.
difficulties. Yeah. If no one have a question yet, I have a few. Oh, Lenaik, I see Lenaik have a question. You can start maybe.
Well, thank you very much, Anna. Very nice talk, very interesting.
Thank you.
Well, yeah, it's very interesting. We have the same kind of vertical walls in canyons in the Bay of Biscay, looking very much the same as the one you have. And we are doing the kind of same analysis that you are doing, and we were wondering, we are struggling about how to choose the right resolution for the 3D model. And I was wondering, you have been using a very low resolution of about 50 centimeter, which is about the size of the sponge or corals. And so I was wondering whether the structure created by the species might influence the terrain variables that you are using. Is it something you have been looking at?
Yes, I have been looking at that. Actually, I noticed that because if you think about the 3D reconstruction, as closer as you are to the substrate, as more as you have the base of the coral there. So what I did in order to try to not have this problem is, first of all, we have explored all these different scales. It's not that I didn't explore all the scales, so I start with 2, 5, 10, and all the scales possible. And actually, I noticed that finer scale have more problems with this rugosity caused by the organisms. So actually going to the larger scale, in my case, helped more because the species that I was studying, they were not so big. So for example, as you can see, in my case, it was for the coral, it was the orientation, one of the most important variables. And the orientation, a very, very tiny, fine scale, if I could have used 10 centimeters, that would have been the coral itself. But as I used the 50 centimeters, then I could capture more the terrain variability of the vertical wall. But I think in the case of vertical wall, it's a very particular case and will change from one to another study and the type of species that you are studying. So it will be very important to consider your species before and do some trial failure before you move forward with the analysis, because it can take some time to get there.
Okay, thank you very much.
Thank you for the question.
Oh, Loic, I have a question.
Thank you, Anna, for this great presentation. I have so you disentangle niche partitioning based on topographic variables, right? Well, I'll turn on my camera. And what about competition? So, for example, if you have corals that are sensitive, let's say, to the proximity of sponges, is this something that you could test, or something that would be doable to test actually with photogrammetry, or is it a limit of the study?
Competition, if you can look at competition between species. So the basis of the niche analysis that there is actually competition. So niche differentiation is based on the species trying to avoid that to happen, because if there is competition, so they cannot be coexistence of the species. So it was interesting to see that they actually using the environment in one or another way to eliminate this competition. But we can, if we want to look at competition, I'm not sure if we could use photogrammetry for that cause, because normally the studies that look at competition need to, first of all, we should have the ability to come back and see a very fine scale how the growth of a coral is affecting the growth of a sponge. So probably for shallow water coral reefs, that will work very well. I'm not sure for deep sea, will be quite expensive. You have to do repetitive measurements in order to see this overgrowth, for example, of corals and sponges. So that will be the my view on that.
Thank you. I'm thinking of one paper, Mitchell and Harris 2020, who does that. In case you're interested, they do that with growth and mortality, but mortality is a different one for you kids because probably you don't see more dead organisms, obviously. But thank you for your answer.
Thank you so much. Yeah, and I'm very happy to have the paper.
Yeah, I'll send it to you.
Yeah, I also had a question that will follow what Loic was asking. So you showed 3 different OMI graph analysis, and does it correspond to different vertical wall or different area?
Different vertical walls. We have
Yes.
3 different vertical walls.
Exactly.
So, yeah.
So I was wondering, because it's not always the case, but sometimes you have the same species in different vertical wall. And if you take like the overall data set and you compare the niche of the species between different vertical wall, I think it could be interesting to see like between different wall, do you have the same niche for the same species, the morpho species, or is it variable? And how? So for you only have three wall, but how is it evolving depending on the wall? Like, do they and depending on the other species present? For instance, in one wall, like if you have like a sponge and on the other wall you don't have a sponge, do you see difference for the niche of one coral, for instance, you know? I don't know if you followed.
So let me see if I understood your question. You're mentioning if I would have, first of all, same species in 2 different vertical walls, and if I will find the same tendency and preference for variables. Actually, I was very happy to find that in the wall. So for Solenosmilia, although we didn't have the same, well, in one wall we have Solenosmilia and Herwigia, and the other we have Solenosmilia with Geodia and the other globular sponge. But then Solenosmilia in both walls consistently show preference for the orientation.
Okay.
So we did observe that. It would be ideal if I would have another wall that have enough presence of Geodia instead of Herwigia, so we can have the same exact comparison. But as Geodia and Herwigia, they are at least sponges, and we did see this preference for the orientation in this case. So that was good. Yeah, and something that is very interesting to mention here is like this, I don't know if interesting, but important, is that this research was, and all these 3 reconstructions were reconstructed with data that was collected opportunistically. So this was a geological survey. I would like to be able to do this type of research with a more, if we from the beginning come with the idea of do this type of research, so we could make sure that we have 2 replicates on the same wall, will help a bit. But anyway, niche differentiation will be a study at the scale of 1 wall.
Okay. But yeah, I think for like comparing for a single species or the same species between wall, like it would be interesting to look if the niche are staying the same or evolving, and then comparing with the literature for what we know.
Thank you. Yeah, I think so too.
And regarding the sponge that share the same niche, because it's only morpho species, do we have an overall good idea of the diversity of species present there, or like because I guess there could be some cryptic species for a single morpho species. So
Yeah, so we
how
didn't
would
go
you
to a level of species, as you could see. I had to go
Mm.
to a morph-species level because we have the limitation of video,
or
which
imagery?
is common.
Mm.
Yeah, which is common in deep sea research. So we couldn't go to a. I think if we would be able to go to a species level, would be better because probably they will even find more differences between species. Now, even this high overlap between the niche of round sponges and geodia, I mean, it also might be because they are very similar, and we don't really probably if I will divide the globular sponge by species, we will know which species exactly they are. There might be more differences between them that I could observe with the data I had.
Yeah, or also like they could differ, have a niche differentiation in another aspect. For example, like the trophic, like they have preference for the same type of habitat, but they eat completely different food source.
Yes.
Then,
Yeah, and there is some research in terms of trophic web for corals and sponges. Yeah.
Pretty cool.
Yeah, and also we have to see that here we have only one part of the niche. We are talking only about variables. This will be very helpful to have extra information such as nutrients, concentration, all that. But what is interesting is we didn't have to go and collect more data, which can be very expensive and time consuming. And so from what we had, we were able to look a little bit deeper into the relationship with this species, and so use these 3D reconstructions and photogrammetry techniques for answering ecological questions. That might be helpful for important problems such as coral restoration, which is so expensive. And there are more governments investing on that, so it will be interesting where we at least know what the species are searching for better than we do so far.
Well, it's already a very good and impressive data set.
Thank you.
And does anyone else have a question for Loic or for Anna?
Yeah, for Anna, considering that these species are kind of immobile, if there were like higher sedimentation rates or some type of thread, would you expect the location to be different?
Yeah, so I could hear my voice for some reason. So thank you for the question, Laurentina. I actually wrote about that in my dissertation because I also found that interesting. And although we don't have much research about this selection in corals, in deep sea corals, we do have it for shallow water corals. So shallow water corals, they seem to, corals in general seem to be animals that are just, they are sessile for sure, they are. But in the phase of when they are larvae, they actively search for areas, and there is a research in shallow water corals that show that they actually can show preference at centimetric scale for one or another type of substrate. And so that is important to notice. But then if the species is there because there are other species close by, because these species also are clones of each other, of course they will first be able to occupy this space. But if it is not favorable for them, if there is not currents that favor their presence, they might not be able to sustain their population in those areas. They might only be able to sustain the population where is their niche. And so that's how those deeper ecological dynamics might happen in the deep sea. But in order to answer at this very specific level of how the species might be doing it, we will need to do a lot more lab work, which is, there is some advance on that, but we still have a long run. I see. Thank you. So that's my answer.
Thank you.
Loic?
I see. Yeah, Loic is raising.
Interesting enough, I discovered recently in the abyssal plane, so it's not on vertical wall, but some sponges are actually able to move. So I don't know if it's the case in vertical wall, but you know, they can maybe move to, if they move, they could find their habitat actually
They move,
based
yeah.
on that. Yeah.
Oh.
Based on time lapse. Yeah.
Yeah.
So that's
what Tina mentioned, that I present this in Malaysia, and she was mentioning that I couldn't find that research. So you could share that with me. I would love to have that.
yeah. Give me a second.
Thank you. Tina Molostova mentioned that she attended this presentation, well, when I had preliminary results, and she mentioned that actually corals could move when they are adult. Like, okay, I was
Corals, I
searching
don't
for
know.
it. So what did you mention? No corals?
Sponges.
Ah, sponges. Yeah, sponges can move for sure, but well, not really. Can you share that with me, please?
I think it's on the chat.
Great. Thank you.
I put the references I mentioned on the chat.
Yeah. Thanks a lot, Loic.
Okay. Thank you, Loic. It's great.
Unless anyone else have a question, I think we're gonna close the seminar. Thanks everyone for joining. It was a really great presentation and fruitful discussion, I think. And so currently we don't have any seminar schedule for the moment, but if any of you, or if you know any student that would be interested to share their work, they can reach us, and we will organize a new session of seminar. Also for the news related to the Deep Sea Biology Society, the SBS 17 is approaching in Hong Kong, and the website is open. If we have not shared it yet, we will share the link to the website of the conference very soon with all of you. And the abstract submission and the registration are already open, so you can start planning if you are joining the conference there. So if I don't see you at a future seminar this year, maybe we will see all of you at the conference in Hong Kong. Okay. Good evening, or good morning, good end of morning, everyone, depending on where you are, or good night if you're in the middle of the night and you're a night owl. And yeah, see you next time.
Thank you, Pierre.
Thank